yeah. Nah, you niggas be capping a lot.
Good afternoon, sir. It is my report for forensic chemistry and toxicology. It is all about blood and blood stains. The pattern of a stain and the quality of blood present can be important close to the nature of the accident of, or crime. Moreover, details analysis of the blood obtained from a stain can reveal genetic and other information that can help identify a victim or implicate the person responsible. It is the importance of blood and blood stains to a crime investigation. Properly collected and preserved blood evidence can establish a strong link between an individual and criminal act, blood evidence, or the lack of blood evidence can also be used to bolster or contradict a witness statement or any statements that the suspect may make. Why are blood stains used for forensic investigation? Because the blood behaves according to certain scientific principles. Trained blood stain patterns analysts can examine the blood evidence left behind and draw conclusions as to how the blood may have been shed. Blood is constantly circulating fluid providing the body with nutrition, oxygen, and waste removal. Blood is most liquid with numerous cells and proteins suspended in it, making blood thicker than pure water. The average person has about 5 liters more than a gallon of blood. And it is the composition of blood. It has four main components: plasma, red blood cells, white blood cells, and platelets. Blood has many different functions, including transporting oxygen and nutrients to the lungs and tissues, forming blood clots to prevent excess blood loss. And it is the platelets cause blood form, damage vessel to clots, and the white blood cells is protect the body from infection, foreign proteins, and foreign bodies. And the red blood cells carry oxygen and carbon dioxide. They contain protein called hemoglobin, which gives them a red color. And the blood plasma composition. Fibrinogen help form blood clots to halt bleeding. It is one of several coagulation factors. Albumin maintains colloid osmotic pressure and transports some substances. Globulins help transport substances and determine an organism's immune properties, other components. Some proteins and enzymes, water, electrolytes, Na, Ce2Cl, and K, Mg2, HCOH, hormones, vitamins, oxygen, and carbon dioxide, coagulation factors.
Blood is responsible for transporting oxygen to the cells and removing carbon dioxide for excretion by the lungs. Blood carries nutrients, ions, water, hormones, and enzymes to all body cells and carries away the waste products. Blood regulates the pH and temperature of the body. Blood is composed of the liquid portion, plasma, and solids called formed elements. Plasma makes up 55% of the total blood volume, 91% which is water. Plasma proteins are albumin to maintain osmotic pressure and water balance, globulin antibodies to help the immune system, and fibrinogen, an essential component for blood clotting. The formed elements are erythrocytes or red blood cells, leukocytes or white blood cells, and thrombocytes or platelets. Red blood cells have no nucleus, therefore they do not divide and only live 120 days. Red blood cells contain hemoglobin that carries oxygen to all cells of the body and then carries away carbon dioxide. There are 5 types of white blood cells, 3 granular and 2 non-granular. Under the microscope, the granular types appear to have grains. They are neutrophils, eosinophils, basophils. Neutrophils phagocytize or eat up foreign substance and secrete lysosomes that destroy bacteria. Eosinophils produce antihistamines that combat irritants. Basophils are also involved in allergic reactions, secreting heparin, histamines, and serotonin. Monocytes also phagocytize bacteria and dead cells, and when they enter the tissue they are called macrophages. Lymphocytes produce antibodies and are crucial to the body's immune system. Bacteria are being destroyed by neutrophils, lymphocytes, and macrophages. Platelets initiate the chain reaction in clot formation.
Plasma is a light yellow liquid. Plasma carried water, salts, and enzymes. The main plasma is to take nutrients, hormones, and proteins to the body that needs. Its cells also put their waste products into the plasma. And the red blood cells, a type of blood cells that is made in the bones, marrow, and found in the blood. Red blood cell contain a protein called hemoglobin, which carries oxygen from the lungs to all parts of the body. Checking the number of red blood cells in the blood is usually part of the complete blood cells (CBC) test. It may be used to look conditions such as anemia, hydration, malnutrition, and leukemia, also called erythrocyte and RCB. And the number 3 is white blood cells. White blood cells are part of the body's immune system. They help the body fight infection and other disease. Types of white blood cells are granulocytes, neutrophils, eosinophils, and basophils, monocytes and lymphocytes, T cells and B cells. And the number 4 is blood platelets. Platelets are thrombocytes, are small colorless cell fragments in our body, in our blood, that forms clots and stop prevent bleeding. Platelets are made in our bone marrow. The sponge-like tissue inside bone marrow contains stem cells that develop into red blood cells, white blood cells, and platelets.
We study about blood disorders, we must know the composition of blood in a healthy individual. Blood is a body fluid in humans and other animals that delivers necessary substances such as nutrients and oxygen to the cells. In vertebrates, it is composed of blood cells suspended in blood plasma. The blood cells are mainly red blood cells, also called RBCs or erythrocytes, white blood cells, also called WBCs or leukocytes, and platelets. Plasma, which constitutes 55% of blood fluid, is mostly water, 92% by volume, and contains dissipated proteins, glucose, mineral ions, hormones, and carbon dioxide. All the blood cells families work together and maintain your body so that you are healthy and strong. Red blood cells transport oxygen to your body's organs and tissues. White blood cells help your body fight infections. Platelets are tiny molecules that stick up together and build a clot on bleeding wounds. All three cell types form in the bone marrow, which is the soft tissue inside your bones. Your body needs millions of blood cells produced by bone marrow to carry out vital functions. But sometimes bone marrow cannot work properly due to certain factors and results in blood cell disorders. A blood cell disorder is a condition in which there's a problem with your red blood cells, white blood cells, or the smaller circulating cells called platelets. Blood cell disorders impair the formation and function of one or more of these types of blood cells. Thalassemia is a group of inherited blood disorders. These disorders are caused by genetic mutations that prevent the normal production of hemoglobin. Hemoglobin is the carrier molecule which carries the oxygen obtained during the process of respiration. When red blood cells do not have enough hemoglobin, oxygen doesn't get to all parts of the body. Organs then do not function properly. These disorders can result in bone deformities, enlarged spleen, heart problems, growth and developmental delays in children. People who suffer from thalassemia need to undergo regular blood transfusions to maintain healthy RBCs and hemoglobin levels in the body. Leukemia. Leukemia is blood cancer in which malignant white blood cells multiply inside your body's bone marrow. Usually, leukemia involves the production of abnormal white blood cells, the cells responsible for fighting infection. However, the abnormal cells in leukemia do not function in the same way as normal white blood cells. The leukemia cells continue to grow and divide, eventually crowding out the normal blood cells. The end result is that it becomes difficult for the body to fight infections, control bleeding, and transport oxygen. What causes leukemia? Is leukemia hereditary? The exact cause of leukemia is not known, but it is thought to involve a combination of genetic and environmental factors. Leukemia cells have acquired mutations in their DNA that cause them to grow abnormally and lose functions of typical white blood cells. It is not clear what causes these mutations to occur. One type of change in the cell's DNA that is common in leukemia is known as chromosome translocation. In this process, a portion of one chromosome breaks off and attaches to a different chromosome. Most cases of leukemia are not believed to be hereditary, but certain genetic mutations and conditions can be passed along to offspring that increase the chances of developing leukemia. Treatments. Treatment for your leukemia depends on many factors. Your doctor determines your leukemia treatment based on your age and overall health, the type of leukemia you have, and whether it has spread to other parts of your body, including the central nervous system. Common treatments used to fight leukemia include stem cell transplant, chemotherapy, and radiation therapy.
Important of the study bloods. In blood group, serology, a number of recent discoveries is of immense value in modern scientific knowledge. Forensic application of blood group studies is a great value in detection of crime. Determination of paternity is another feature in forensic medicine after studying the blood group serology. Number one is as a circumstantial or corroborative evidence against in favor of the perpetrator. Number two is as evidence in a case of disputed evidence. The number three is as evidence in the determination of the cause of death and length of victim survival of attack. And the number four is as evidence in the determination of the origin, determination of the direction of the case of the victims or the assailant. The number five is as evidence in the determination of the origin of flow of blood. The last one is number six as evidence in the determination of the approximate time the crime was committed. There are the four chronologicalities for blood. Number one is preliminary test. Preliminary test, also known as preliminary test screening test or failed test, establish the possibility that a specific bodily fluid is present, do not conclusive prove the presence of the specific substance. The number two is confirmatory test. Confirmatory test for blood include identification of blood cells under a microscopic cellular 2002 crystal tests such as the Tickman and Takayama test, cellular 2002, expanding 2003, and ultraviolet absorption test, Genslin 1983. And the number 3 is precipitin test. The precipitin reaction is also known as the biological reaction for proteins. It enables us to distinguish between protein by the animal body as a test tube and to establish differences between them which no other form of test tube will detect. It is best known as a means for distinguish human from other forms of blood. The procedure is briefly to inject an animal, usually a rabbit, repeatedly with a foreign protein. The rhizome of that animals that gives a precipitate with that proteins but with no other. So if the materials injected is human blood, is precipitate is used produce when the rhizome of the blood of the rabbits added human, and with any other sort of bloods. The reaction of its value is forensic medicine, and it is also of values is the zoologist. It is enable him to demonstrate the blood relationships of animals and by the amount of precipitate to ascertain the degree of the relationship is in figures. And the number 4 is blood grouping test. The test to determine your blood group is called about types. Your blood sample is mixed with antibodies against type A and B blood, then the sample is checked to the whether or not the blood cells stick together. If blood cells stick together, it means the blood reacted with one of the antibodies. Preliminary test for blood. Number 1 is letter A, benzidine test. Benzidine test is most commonly used for the detection of blood in the feces and was therefore singled out for investigation. Result in intense blue colors produced immediately. And the letter B is phenolphthalein test. Is a presumptive test that react with the heme molecule present in blood. A positive reaction gives a pink color, while blood stain normally appear red-brown in color. The color of substrate or the edge of a stain may affect the appearance or visibility of the stain. Result: rose color develop deep pink permanganate color. And the letter C is the guaiacum test. It is the fecal occult blood test. VOBT is used to find occult blood or blood that can be seen with the naked eyes. To stall the idea behind that this test is blood vessels at the surface of larger polyps or cancer or are often fragile and easy damage by passing stall and result beautiful blue color that appear immediately. And the letter D is the leukomalacyte test. The leukomalacyte grain presumptive test for blood is a catalytic test which is based on the peroxidase-like activity of hemoglobin. Hemoglobin has the ability to cleave oxygen molecules from H2O2 and catalyze the reactions from the reduced form of leukomalacyte grain to the oxidized blue-green colored product. And the letter D and the letter E is the luminol test. The luminol test chemiluminescence reaction is responsible for the glow of latex. The reaction is used by criminalists to detect traces of blood at crime scenes. In the test, luminol powder C8H7O3N3 is mixed with hydrogen peroxide H2O2 and hydroxide Ag KOH in a spray bottle. Result: luminescence or emissions of light. Confirmatory test for bloods. Confirmatory test for blood. The actual proof that a stain is blood consists of establishing the presence of the characteristic of blood pigments, hemoglobin, or one of its derivatives. Hemoglobin is the red coloring matters of the red blood cells of the blood. Under the three confirmatory tests for blood that determines whether satin is really blood are: number one is the microscopic, number two is my microchemical test or microcrystalline test, and the number three is spectroscopic test. Microscopic test for a blood smear test. A laboratory professional examines the slide under a microscope and looks at the size, shape, and number of different types of blood cells. These include red blood cells, which carry oxygen from your lungs to the rest of your body, and white blood cells, which fight infection. Microscopic examinations, which identifies and counts the types of cells, cause cholesterols, and other components such as bacteria and mucus that can be present in urine. And the microchemical test or microcrystalline test. Microcrystalline tests are chemical tests resulting in the formation of unique microcrystals for a given substance when combined with a specific reagent. Microcrystals are observed under a microscope, and micrographs constitute the result of the test. Spectroscopic test. As a spectroscopic test method I described, whereby the percentage oxygen saturation of hemoglobin within intact corpuscles can be determined. A complete oxygen dissociation curve can be determined with 1 to 2 cc of whole blood. The microchemical test and microcrystalline test for blood. The identification of blood can be made more specific if microchemical or microcrystalline test is applied. or perform Takayama test and Tickman test are the most popular ones. There are the three: microchemical and microcrystalline for blood. Letter A is Tickman. Letter B is the acetone test. Letter C is hemochromogen crystal test. The Tickman test is the confirmatory test for blood based on the formation of distinctive hematine crystal that are viewed under a microscope. The reagents typically used are sodium chloride and glacial acetic acid. Result: dark brown rhombic crystal of hymine or hematine chloride, arranged singly or in cluster. And the letter B is the acetone test. The acetone test is a ketones in blood test measure the level of ketones in your blood. Ketones are substances that your body makes if your cells don't get enough glucose, blood sugar. Glucose is your body main source of energy. The result is small dark diachronic acicular crystal of acetone hymine, and there is a hemochromogen crystal test. Hemochromogen crystal information with minutes amount of blood. A method is described whereby small amounts of blood, such as residual materials from the pre-bitten test, may be used positively to indicate the presence of blood via the formation of pyridine hemochromogen crystals. Result: large rhombic crystals of a salmon pink arranged in cluster. Sieves and other forms that appear within 1 to 6 minutes when viewed under the lowest power project cover with glass slip. The spectroscopic test for blood. Spectroscopic method is described whereby the percentage oxygen saturation of hemoglobin within intact corpuscles can be determined. A complete oxygen dissociation curve can be determined with 1 to 2 cc of whole blood. The spectroscopy is a class of tests which study the interactions of samples, specimens with energy, usually the perform of light. Results: upon absorbing some of the rays from the spectrum, it produces characteristic dark colors bands which vary with the types of blood pigments. Sa blood group inherence. Blood group are inherent from the both the ABO blood type is controlled by a single gene. The ABO gene with three types of alleles infers from classical genetics. I IA and IB, as both IA and IB are dominant over I only II. People have type O blood. There are 11 terms to founders. Number 1 is gene. Gene is the basic physical functional of unit of heredity. Genes are made up of DNA. Some genes act as instructions to make molecules called protein. Every person has two copies of its gene, one inherit from its other. The basic unit of heredity that occupies a specific location on a chromosome. It consists of nucleotides arranged a linear manner. Most genes code for a specific protein or segments of protein leading to a particular characteristics or function. The number 2 is chromosomes. Chromosomes are thread-like structures located inside the nucleus of animals and plant cells. Each chromosome is made of proteins and a single molecules of deoxyribonucleic acid. DNA passed from parents to offspring. DNA contains the specific instructions that make its type of living creature unique. The small bodies in the nucleus of a cell that carry the chemical instructions for reproduction of the cell. The consists of strands of DNA swarm in a double helix around a core of proteins. Each species of plants or animals has a characteristic number of chromosomes. The number 3 is phenotype. The term phenotype refers to observable physical properties of an organisms. These include the organism's appearance, development, and behavior and organisms. Phenotype is determined by its genotype, which is the set of genes the organism carries, as well as by environmental influences upon this gene. Phenotype is an individual observable traits such as height, eye color, and blood type. The genetic contribution to the phenotype is called the genotype. Some traits are largely determined by the genotype, while other traits largely determined by environmental factors. The number 4 is genotype. Genotype is in a broad sense, the term genotype refers to the genetic makeup of an organism. In other words, it describes an organism's complete sets of genes. In a more narrow sense, the term can be used to refer to the alleles or variant forms of a gene that are carried by an organism. Number 5 is the homozygous. The homozygous genotypes or pure genotype, the presence of two identical alleles at a particular gene locus. A homozygous genotype may include two normal alleles or two alleles that have a same variant. The number 6 is heterozygous. Heterozygous genotype or hybrid. Heterozygous refers to having inherited different forms or a particular gene from its parent. A heterozygous genotype stands or contrast to a homozygous genotype, where an individual inherits identical forms of a particular gene from its parents. Number 7 is gamete. Gamete are organisms' reproductive cells. They are also referred to as sex cells. Female gametes are called ova or egg cells, and male gametes are called sperm. Gametes are haploid cells, and each cell carries only one copy of its chromosomes. These cells develop into sperm or ova. And the number 8 is sperm cell or microgamete, male sexual cell. And the number 9 is egg cell or microgamete, male sexual cell. And the number 10 is zygote. Zygote, fertilized egg cell that result from the union of a female gamete, egg or ovum, with a male gamete sperm in the embryonic development of humans and other animals. The zygote stage is brief and is followed by cleavage when the single cell becomes subdivided into smaller cells. The number 11 is alleles. The alleles, an allele is a variant form of a gene. Some genes have a variety of different forms, which are located at the same position or genetic locus on a chromosomes. Humans are called diploid organisms because they have two alleles at its genetic locus, with one allele inherited from its parents.
Human blood has two major components, namely blood cells and blood plasma. The membrane of the RBC has two types of proteins called antigen A and antigen B. The plasma contains two types of proteins called antibody A and antibody B. The four types of blood groups are A, B, AB, and O blood groups. These letters refer to the antigen present in the red blood cell. That is, A group has antigen A, B group has antigen B, AB group has both the antigens A and B, while O group has no antigens in the red blood cells. The antibodies A and B in the plasma can react with corresponding antigens and coagulate the blood. So plasma of A group blood will have only B antibody. In B group blood, A antibody is present. AB group has no antibody, while O group has both A and B. The reaction between antigen and antibody has a lot of significance in medical science. Before any blood transfusion, doctors make sure that the blood groups of the persons involved are compatible. Let us see how this happens. Take 2 drops of blood from an individual with any blood group. To each of these drops of blood, add a drop of antibody A or antibody B. What do you expect to notice? If the blood drop coagulates with antibody A and not with B, it is group A. If the coagulation is with antibody B, it is group B blood. If coagulation is caused by both the antibodies, it is group AB blood. And if there is no coagulation, it is O group blood. Conclusion. The blood group testing is based on the property that in any individual, the antigens and antibodies are never the same, because antibody A agglutinates antigen A. Similarly, the antibody B agglutinates antigen B.